Okay. Um, well, it's an absolute honor to be the very first speaker in what hopefully is a going to be a long-standing tradition of ASP's Natural Product Sciences webinar. Um, Now, before we get going, I want to make sure everything's working properly here since it's our first time. Everybody just touch your screen to make sure you can advance the slides. Okay, maybe touch and swipe. Okay, I'm just playing actually. Touching the screen doesn't advance the slides. Okay, let's get started. My name is Brian Murphy. I'm an associate professor at the Department of Pharmaceutical Sciences at the University of Illinois at Chicago. And my talk today is called Toward Antibiotic Discovery from a Bacterial Colony. And I added this subtitle, A Primer for Academia, because what we're really trying to do here is build methods that are suitable for those operating on smaller academic budgets. And I hope that there are several useful takeaways as to how, in particular, young scientists might be able to form a competitive drug discovery program for microbes. So here, this is one pretty standard version of the pipeline. So you have, if you want to discover new natural products from the environment, you can go out and do sample collection. Our lab does a lot of scuba and boat collections. You then generate microbial libraries. And then after that, you're going to use these microbial libraries to generate natural product fraction libraries. You then add these fractions to a series of biological screens. You do lead dereplication to find known natural products, and then you do lead identification of any potentially new natural products. Now, compared to other steps downstream, the philosophy that we use to collect samples from the environment, and especially to generate microbial libraries, this has really seen few widely applied conceptual changes in nearly 8 decades of biomedical research. And that's what we're going to focus on in the talk today, improving these steps. Now, of course, one of the main problems in this pipeline is that there ends up being a high degree of both taxonomic redundancy and natural product redundancy in microbial libraries that are sourced for drug discovery, and there's a lot of downstream deleterious effects, including the rediscovery of known drug leads. It's an inefficient process, a very costly process, and as a result, there's in some cases an emotional divestment, in a lot of cases a financial divestment, as we've seen in companies divesting from the, in particular, antibiotic discovery process. And I think these limitations really become clear when you're looking at a single bacterial diversity plate. So even if you employ an expert microbiologist to pick colonies and to build your library, you're really still limited to either morphology or at best 16S ribosomal RNA gene sequence data or something similar. And the important point is that these are not necessarily indicative of the natural product producing potential here. And so what we really want to know is how much overlap is there between similarly looking colonies? Do these produce the same natural product populations? Do they produce significantly different natural product populations? Because it's this type of information that we're really going to use to build our library. And this library is going to be what our graduate students use and what our collaborators get a hold of. And the more redundancy you have, the more the downstream processes are going to cost more and be inefficient. And so I guess one of the main goals of my lab is to turn this process basically into something that looks a little bit more like this. Now notice we cut out a step here, natural products fraction library generation. What my lab aims to do is perform as many of these steps as possible on a single bacterial colony. Now in turn, this is going to speed up the process, make it more accessible to academic labs that have fewer resources, and hopefully it'll cost less and be more efficient. Now this isn't to say that we're not arguing against creating a fraction library for natural products. That has its advantages as well. This is just one other way to do it, and I'm really I'm really focused on trying to extract as much information as I can from a single bacterial colony. Okay, and the way that we're going to accomplish this is through our collaborator, Dr. Laura Sanchez. Dr. Sanchez is an expert in MALDI-TOF mass spectrometry, among other topics in natural products. And here's a really brief overview of the process. You have your sample on a plate, you coat it with matrix, you hit it with energy, molecules ionize, desorb, and then travel through this time-of-flight tube. And you eventually the ions hit a detector and you generate a mass spectrum. And for the remainder of the talk, I'll be referring to this mass spectrum as a fingerprint, because that's kind of how we're going to use it. Now, the sample preparation for this is very easy. You have a sterile toothpick, you get some cellular material, so by stabbing the colony, you smear a small amount of those cells onto a MALDI target plate, steel MALDI target plate, and then you add a microliter of formic acid and a microliter of matrix, and you're ready to run your analysis. So to date, this is what's been done, and I listed a couple review articles down here below. To date, this technology has mostly been used in hospitals, so things like Bruker Biotyper or bioMaru have analyzed the protein, the MALDI protein mass spectrum, 3,000 to 15,000 Daltons, and basically generated fingerprints that they compare against a database of clinical isolates. And so these fingerprints, you look for a match, and then you can get subspecies level resolution. So what we intend to do, the way we're going to innovate this process is add a whole nother layer. Not only are we going to acquire a protein mass spectrum of each colony, but we're going to acquire a natural product spectrum of each colony. And the information that we get in return is that from one, we answer the question, who are you? All right, what type of bacterium, genus, species, maybe even subspecies of bacteria? And the second question is, of course, what do you produce? And it's these two questions that I think are fundamental to building a diverse, minimally overlapping library that we source for drug discovery. And you have to realize that this is just one single plate here. All right, now traditionally people have just used morphology and 16S to select colonies here and there. But what we propose to do is to pick every single colony on every single plate. And in a lot of cases, we have hundreds of plates and thousands and maybe even tens to thousands of colonies. And this is one major way that our process is going to differ from what's known. And I think the important part is that it removes a lot of bias from the discovery process that I think has hindered our ability to discover new drugs. And I'll get into that in a little bit later. So in order to handle all these data, because now we're going to be generating two different spectra for every single bacterial colony, we had to build a bioinformatics pipeline to process all these data. And my student Chase, very talented chemist and computer programmer, designed IDBack. So this is freely available online. You can download it at the link here. And I'll briefly go over how this works. I think IDBack can be summed up in three of our publications. The first one, we really were just reporting the abilities and limitations of IDBack. The second publication, because it's freely available, we wanted to give people a really nice tutorial on how to use it. And the third publication is the application of IDBack to drug discovery. How to build a library that has minimal taxonomic and natural product redundancy in it from one large-scale collection trip. And I'll give one or two brief slides on that. But first, how the process works. You have a bacterial diversity plate, and we're going to isolate each one of these colonies. Now, importantly, we're not going to run these analyses directly off of the plate because there could be crosstalk, intra or interspecies crosstalk, that really affects natural product production between the colonies. So two seemingly identical colonies, such as 2 and 11, maybe they're identical, but because they're in different environments on the agar plate, the natural product production would seem different. So it's very important we do this step. We isolate each individual colony, get it growing in the same media, on the same time scale, into individual wells of a multi-well plate. And it's from here that we do the analysis. So after acquiring the data, when we look at our protein spectra, these data are organized into a dendrogram. And this is where you can see similar colonies grouping together. The question, who are you? So the orange colonies are all Rhodococcus strains, and they all group together. They have high similarity. What's really interesting now is that we can look at what we call a metabolite association network. This bottom network here, and this is the natural product region. These are your bigger nodes are your bacterial colonies. Your smaller nodes are your M over Z values, or natural products. And what's interesting here is you can look at your Rhodococcus colonies and see that there is basically a heavy degree of overlap of natural product production. Yet, when you look at these two Penibacillus strains that are indistinguishable on a plate, and they have greater than 99% 16S similarity, of course there's an overlapping natural product population, but there are also unique nodes, right, that are unique to one colony over another. Well, this is information that you want when you're creating a library. We believe these are peptides between 1,000 and 1,200, and even some up to 1,500 M over Z values. And this is an important distinction between these two, because now we can make an informed decision about what kind of chemistry goes into our library, and we can not add particular strains to it. And so we performed this on the scale of a single collection trip. This is the work of Dr. Sofia Costa, previous graduate student of mine, and Dr. Cecilia Omarsatir from the University of Iceland. And Chase was also a co-author on this work. So we went on 5 different expeditions to Iceland, and we picked the latest one and used that as an example to try to create what we call a smart library. Now here's a dendrogram from all of the isolates. There were 1,616 isolates in total. After analyzing all of these and analyzing the metabolite association networks of each of these different pseudo-phylogenetic groupings, we added 302 strains to the library. Now, because of our untargeted colony picking practices, because we chose to pick every single colony on every single plate and not just select ones that looked good by our eyeball, we ended up getting from these 102 isolates greater than 65 genera represented. And we think this is a real testament to the effectiveness of this method to access natural products and taxonomic diversity. And importantly, 1,300 isolates were not added to the library, so we don't have to deal with this overlap in downstream processes. Now, if you'll allow a brief aside, and I promise that this relates directly to Sophia's work, at the same time, my students, Dr. Mary Malfecchi, who she recently defended, she asked a very simple question. And this was in collaboration with Dr. Nadine Zeimert's lab at the University of Tübingen. From 2 sediment samples that we collected in Lake Huron, as part of the Great Lakes in the United States, how much natural product capacity, and by capacity we mean the presence of specific classes of biosynthetic gene clusters in sediment and on plates, how much of this capacity are we recovering when we use just standard cultivation techniques and several types of common nutrient media? Now, in the paper, she details several findings and limitations, but one finding that was that the majority of biosynthetic gene clusters from readily cultivatable bacteria were either uncharacterized or not deposited in known gene databases. So of course, these databases aren't complete, but this still indicates that some genes on plates harbor potential to produce new natural products. But this kind of contradicts what we typically see from standard cultivation approaches, right? We see that there's a high probability of discovering known chemistry. So what's up with this disconnect? Why this disconnect? Well, we kind of hypothesized that for decades the field has been using similar biased approaches to colony picking, and they've been focusing heavily on spore-forming bacteria like actinobacteria or bacillus. And we think that there's many understudied isolates that are sitting here right on these plates that are ripe for discovery, and we can actually show some data to support this. So back to Sophia's dendrogram, you can see each of these are different microbial isolates, but when you zoom in on this little region here inside the box, these are some actinobacteria that I term understudied. Other people might call them rare actinobacteria, but I take issue with that term. I don't think it's an accurate way to say it. So these are a bunch of understudied actinomycete genera, and in parentheses is the number of publications that report a natural product. So again, we didn't use any special media types, and we didn't use any special isolation techniques or cultivation techniques. This is all standard media types, and these were just sitting there right on the plate. And so, as you can see, we've even got potentially new genera that are visible with the naked eye. And so, if your goal is to target understudied bacteria, which is this is a method that I do recommend, especially for a lot of young professors trying to get into the field. I think that this can be a really nice way to go in and find some of these understudied bacteria. And what's really important also is that when you look at some of these are translated onto a plate now. They don't, quote, look like what you would expect actinomycetes to look like, right? These are, look at, they're slimy. They look like more like gram-negative unicellular bacteria and a lot of types of bacteria that have been mostly ignored in the process. And so it was no surprise to us that when you did isolate all these colonies that you end up getting really interesting genera. Okay, so finally, one other thing that we're trying to do with IDBac is we're trying to remove one last component of human bias from this process, and we're trying to automate the process of library creation. So here's the process. We first group isolates using hierarchical clustering of their protein spectra, and then within each of these groups, Chase designed an algorithm that iteratively selects isolates until a specified percentage of natural product peaks are captured within each group. So here's your dendrogram. There's about 900 bacterial isolates. This is from his data from freshwater sponge collection in the Great Lakes. Now, as we start moving down this dendrogram or this tree, you'll see red circles at the tips of the tree. These are the selected isolates. Now remember, these isolates, the ones that we're going to add to our library, they weren't selected based off of just the protein spectra, but also by taking into account the small molecule MALDI spectra. So here we basically set a threshold within each group that selects the minimum number of isolates while retaining 75% of all small molecule features within the group. And this is an arbitrary number. You can change 75% to whatever you want. And we're currently comparing manual library creation using IDBac with this automated process, and we're gearing up to publish this soon. Okay, on to just to finish up on we'll briefly go over the second part of my talk. We talked a little bit about sample collection and library generation, but now I'm going to talk about how to do biological screening and lead dereplication from a colony. So, of course, agar-based assays have been around since the early to mid-1900s, and there's been many developments since then. You have your classic agar overlay assay or your intruder assay. You have a couple, we picked a couple examples, 3 and 4, that are more recent in the literature that we thought were pretty innovative. There's the Transwell assay by Pishani, Emily Mevers, John Clardy, and Roberto Coulter's group. This was a really neat way to be able to extract molecules from a bacterium grown in solid agar, but they used this like little liquid medium in a Transwell plate. And then there's this Janus plate that was developed that we really loved the concept of this because this was a plate that had 2 sides to it. And so we wanted to develop a plate that used both 2 sides, and I'll show you why we did this in a second here. So there was a lot of design that went into a 2-sided plate, and we wanted to make it a multi-well plate. So my student, Dr. Jung-Ho Lee, he's got a knack for design, so I set him on this journey 2 years ago, really. And he used this freely available software to design our bioassay plate. Now, Jung-Ho recently gave a 45-minute seminar on all the trials and failures. This plate took us about 2 years to design in 3D print, and it wasn't a simple. We thought it was going to be simple, and of course it wasn't. And it was a brilliant seminar by him. And and just to let you know, I won't go into all the details, but I'll tell you that of the 2 years, we went through probably about 40 iterations of designing the plate, and there's so much that I won't talk about the design of the inside and all the different channels that we had to build to deal with air bubbles and getting agar to stick in the plate. But there was so much design that went into this, and I really want to credit him for that. So there he is, Dr. Zhang Li here, and this is our final product. We call it DAPA, or DAPA, the dual-sided agar plate assay. And here are some of the characteristics that we want to highlight. Now, importantly, this plate is autoclavable, and thus it's reusable. So you get to feel environmentally conscious because you're cutting down on plastics use. You're also saving money because you buy one plate or a few plates, or make one or a few plates, and you can use it for a long time. We still are using ours, and so we don't really know what the limit is, but we've autoclaved it, over 30 or 40 times now, and it's still fine. There's no bending or warping. The plate itself is non-toxic, so you're not leaching any chemicals into your agar, giving you false positives in your antibiotic assays. The dimensions are amenable to automation and thus high throughput screening. And importantly, with the design of this plate, which I'll show you in the next slide, we're avoiding unwanted surface interactions. So it comes. We also had to design this little silicone plug here, and this is how it works. So you have your silicone plug, you pour your agar, you let your agar dry, you remove your plug and inoculate a test bacterium. You let your test bacterium incubate for however many days you want, and this test bacterium produces natural products into the agar. You flip your plate over, so now the bottom is the top, and what's really nice is you have this flat surface from which to work, and this really solves a lot of problems in agar-based bioassays that we've been seeing in the past, well, really century or so. So on this flat surface, you add your pathogen and incubate for however long you need. Now, we've been working with the Institute for Tuberculosis Research to design fluorescently labeled pathogens. So we can use fluorescence to readily visualize growth inhibition. So here are some control strains. You can see that there's a lot of growth, and here are some negative controls where there's no growth. What's also nice about this flat surface is that it allows for an orthogonal detection technique in case fluorescence is being interfered with by produced natural products. And so we add this MTT dye, that's a simple colorimetric assay, to detect growth inhibition. Now, through our colleagues in the ITR, Dr. Scott Franzblau, Sang-Hen Cho, and Rui Ma, we've been able to fluorescently labeled the escape panel, or at least most of the escape panel, and we're currently in the middle of screening this right now. We do have some preliminary results because we really just recently started this. From those bacteria that I showed you from Iceland, we took a subset of 41 isolates and we put them on a plate. So down below we have some controls on the plate. These are ATCC streptomyces strains that are known to produce antibiotics, so they exhibited growth inhibition. But we also saw some growth inhibition within 5 of our isolates. And so we'll focus in on just one. This is K770, a bacillus species. And when you take a little spatula and you simply pop out the agar plugs, that's all. You do that, you grind them up with DCM and methanol, so this is very minimal supplies, little time, and you take that extract and you run LCM SMS analysis, and then you can dump these data into GNPS. This is of course, as we all know, GNPS is the system out of the Dorstein and Banderas labs at UCSD, and this is freely available. So when you pop these data in, we were able to find a near identical match here, and this match was to the cyclic lipopeptide bacillomycin LC2, responsible for the antibiotic activity. Now, what was another cool function is that within GNPS there's a link. It links to NP-Atlas. This is the cool program, freely available also out of Roger Lennington's lab. And we were able to quickly visualize the chemical space of these cyclic peptides because, of course, now you have to make a decision, do I proceed with this project or not? And this NP-Atlas just gives you the ready access to this information. So I think this is a really a nice example of a testament to the work that like the Lennington lab and the Dorsey lab and other labs, maybe even like Marnix's lab from MyBig, where they make these tools freely available online, and it really facilitates research in our group and other labs around the country and world here. And so to have all this information available to us from a simple agar plug is really nice. And now again, I really have hit pause on the discovery program for 4 or 5 years now. But it's exciting times because in the past couple of months, now that we've developed these tools to improve the front end of discovery, we've started to get back into it. So here's just a couple examples. Dr. Lynn Nguyen is a postdoc in my lab right now. She is from the Institute of Marine Biochemistry at the Vietnam Academy of Science and Technology. She's here as part of an NIH program that's going to transition her into an independent faculty position at IMBC in Hanoi. So her and Dr. Tonan Tran were both studying at UIC. Tonan was studying in Alessandro Eustachio's lab, who studies microbial biosynthesis. And they're working together to process this bacterial library from Vietnam. So from 821 isolates, they used IDBAC and MALDI-TOF MS, created a smart library of 267 isolates, and they're currently screening this in the DAPA assay. And they'll bring this work back to Vietnam with them and start their own microbial discovery program. A graduate student I have, this is Antonio Hernandez. We have done a sample collection trip out in the Apostle Islands in the Great Lakes, collected several freshwater sponges, and from 875 isolates, he's got what he thought would capture the natural product space. This is 127 isolates, and currently he's not only using the DAPA assay to do regular screening against the escape panel, but he's also using different elicitors on these plates and using these DAPA plates as a co-culture vessel. And so we're doing a lot of unique things with them because we think these plates are very versatile. And so ongoing future studies, we're in the process with Dr. Franzball and the ITR, we're in the process of purchasing this Rapid Pick Light colony picker from Hudson Robotics. Really awesome colony picker that's able to process I think about 2,500 colonies in 1 hour. So what we're really hoping to do is to basically automate the majority of this front-end process from after we plate the samples, basically from colony picking, because that's what the robot does, to library generation, cryo storage, and even plating on a biological assay. So that's kind of where we're at right now, and hopefully in the next year or two we'll be seeing if this all works, right? That's what the game is. Okay, so I hope that I've been able to convince you that you can do many of these front-end discovery steps from a single bacterial colony. There's obviously limitations to this. This isn't the perfect way. You're missing out on some information by using MALDI. But this is one way to do it, and we think that it really cuts down on a lot of the costs that are associated with generating natural product fraction libraries. And honestly, it was just born out of sheer frustration from some of these older library generation processes that we decided to completely try and rethink how we do discovery. And so with that, of course, I'd like to thank my awesome lab. Here's some of them pictured to the bottom right. Sophia and Chase, who did a lion's share of this work. Jung Ho, Radhika Dakal, I mentioned Lynn and Antonio, several undergraduates who are currently in my lab or formerly in my lab. Dr. Laura Sanchez, who without her, none of this would be possible. She's very collaborative, so feel free to reach out to her and her group members as well. They have been just awesome. All of our collaborators in Vietnam and Iceland, our funding sources, and you for your attention. I thank you very much, and yeah, I'd be happy to answer any questions. And that's it.